Hello everyone. This is our second section of biostatistics, in which last time we discussed about some basic concept of statistics, which include data collection, types of data, then definition of actually biostatistics, then how biostatistics is helpful in biological research. Then we discuss about central tendencies, in which we talk about mean, median, and mode. And at the end, we talk about standard deviation. Okay, when data is deviated from the central value, it is called as standard deviation. So we have 36 different formulas of standard deviation. Now in this lecture,
सर थोडासा आवाज कमी येतोय. आय थिंक माईक जवळ.
हॅलो हॅलो
हा सर, आवाज थोडासा कमी येत होता.
आता येतोय का सर? हॅलो.
हो सर, हो सर.
Okay, so now in this section, I'm going to talk about one of the most important points of biostatistics or test of biostatistics, which is correlation. Most frequently used technique and to our tool, we can say, of biostatistics in biological research, where we can find out the relationship present between two variables. Okay, so I just want to quickly go through the previous lectures point, in that we talk about sampling methods, then types of sampling, then we discuss types of data. In that, three different types of data we have discussed: simple data, ungrouped data, and group data, also called as discrete data and continuous data. Then we talk about characteristics of the group data. And at the end, we focus on measures of central tendencies. Correlation, I'm going to present my screen soon. Now, correlation is the technique which is used in biostatistics to find out the degree of relationship present between two variables. Okay, so with the help of this, you can easily find out how much the relationship is there in between two different variables. Okay, and there are various methods of finding the correlation. Like you can find out the correlation present between the two variable by qualitative approach as well as by quantitative approach. Okay, so you can directly learn about directly find out the degree percent of correlation present between the two variable which you have studied. Okay, so now we're going to directly focus on the correlation part. Give me 1 second. Okay, so the point of discussion for today's lecture is correlation. I'm directly going to start with the actual theory part and what is the significance, followed by formula and interactive problems. Yes, so correlation is the degree of relationship which is present between two variables. For example, you are studying two different variables in your research like X and Y. And if you want to find out whether there is a relationship present between the two variables or not, you can find out with the help of correlation. And there are three important methods by which you can easily calculate the correlation. Okay, so in most of the biological experiment, a certain relationship between the two variables is always present, and in such case
change in one variable lead to change in another variable. Okay, if the two variable are related, then only if variable one changes, then the second will also going to change in some degree. Okay, the change in the second variable due to the change in the first variable is only depend upon degree of correlation which is present between the two variable. Now in correlation studies we measure the strength of relationship present between the two variable. Okay, so there are two different type of correlation studies. Some are qualitative and some are quantitative. In quantitative studies you are directly going to get the value of correlationship present between the two variable, so that it become very easy for you to understand the behavior of two variable in your research. Okay, the statistical tool which help in studying the relationship between these two variable is correlation, and generally we study correlation with the help of these three technique. Like we can say that there are positive or negative correlation. Okay, then simple, partial or multiple correlation, and linear and non-linear correlation. Okay, so there are three important types of correlation. When you study the relation present between the two variable, it is either positive or negative type. In case of positive correlation, if the value of one variable increase, other will also going to increase with it. And if the value of one variable decrease, other variable also going to decrease. It is called as positive. That means both the variable moving in same direction, either increasing or decreasing. In case of negative correlation, exactly opposite thing happen. That means if x increase, y decrease, and if x decrease, y increase. Okay, so we can take multiple examples of positive and negative correlation, like for example input of pesticides or input of fertilizers, say input of fertilizers and crop yield. So it is the positive type of correlation. Okay, for some extent. Okay, initially if you increase the fertilizer from 0 to certain degree, then yield will going to increase. That means both the variable moving in same direction. It is called as positive correlation. But if both are moving in opposite direction, it is called negative correlation. कोणाला सांगता येईल का निगेटिव्हचं एक्झाम्पल एखादं लाईफ सायन्स मधलं किंवा इन लाईफ जनरल मधले पण चालेल. व्हॉट इज मीन बाय निगेटिव्ह? वेन वन इन्क्रीज अदर डिक्रीज, इट इज कॉल्ड एज निगेटिव्ह कोरिलेशन. एखादं एक्झाम्पल, well-known example of negative correlation is when in case of winter, for example, the sale of woolen garments and day temperature. Okay, if the day temperature decreases, garam kapde आपण म्हणतो, woolen garment sale will going to increase, and if the day temperature increase, the sale will going to get decrease. Okay, so it is the example of negative correlation. Many type of negative correlation also present in biological sciences. I'm going to discuss all of them afterward. Okay, so it is called as positive if both the variable moving in same direction, and negative if both are moving in opposite direction. Then another example of or another type of correlation is simple, partial, and multiple. In case of simple correlation, one variable is depend upon another variable. That means only two variable come into the picture. And suppose x is depend upon y, it is called a simple correlation. But in case of partial correlation, one variable depend upon two. Okay, that means more than one variables. It is called as partial. And in multiple correlation, generally we consider multiple variable depend upon one another. It is quite complex type of correlation. Okay, suppose x increases along with x, y, z, and say a, these three variable are also changing depending upon the change in variable x. It is called as multiple correlation. Okay, so simple means the correlation present between the two variable only, partial means more than two, and multiple means many variables. There is a 100% proof of relationship present between the multiple variable. It is called as multiple correlation. And last is linear and non-linear. In case of linear correlation, there is exact degree of change in variable y depending upon change in x. That means x is changed by 10%, and then y will also going to change in similar order. So that when we draw x and y variable on x and y axis of the graph, so now when we consider correlation, there are three types: positive correlation and negative correlation, simple, partial, and multiple correlation, linear and non-linear correlation, right? So in case of positive and negative correlation, if both the variable are moving in same direction. That means if the variable x increase, y also increase. And if variable x decrease, y also decrease. It is called as positive correlation. In case of negative correlation, if one variable increase, other will be decreased. And if the one decrease, other increase, it is called as negative correlation. Okay. Now in nature, you can find out number of example of positive. If the height of plant increase, number of flower will going to increase. Okay. Now if input of fertilizer increase, maximum time yield will be increased. Right. So it will be it is the example of positive correlation. But sometimes you also find negative correlation. Okay. So can anyone tell me the example of negative correlation? Any one example of negative correlation? If both the variable moving in same direction, either increase or decrease, it is positive. But when we
Sir,
can see,
can I give an example?
yeah, yes, ma'am.
The treatment of mutagen and survival of plants.
Okay, so if the concentration or the treatment time increase, the survival will going to get decreased, right?
Yes, sir.
Okay, it is negative. Okay, the treatment time of mutagen and survival rate, it will be a negative correlation, right? Then another very good example is sale of woolen garments and day temperature, right? If the day temperature increases in the summer, the sale of garam kapde म्हणतो आपण, sale of woolen garment will going to get decrease. So it is negative correlation. And if the day temperature decreases, sale increases. So it is negative correlation. Similarly, another type of example is another type of correlation is simple, partial, and multiple correlation. Okay. So in case of simple correlation, you only study the relationship present between two variables, x and y, a or b, a and b. Okay. So if you are studying the relationship present between the two variable only, it is called a simple correlation. But in case of partial or multiple correlation, you are studying the relationship of one variable with more than two variables. Okay, suppose x is increasing, what will be the effect on y and z? It is called as partial and then multiple correlation. Okay, if you consider only two variable in your research, it is simple correlation. And if you are considering more than two variable at a time, then it will be partial or multiple correlation. And the last type of correlation is linear and non-linear correlation. In case of linear correlation, if you draw the line or draw the graph, and if you take the variable on x and y axis, then it will be a straight line. Perfect relation, perfect correlation. If the perfect correlation is present between the two variable, the line will be straight. It is called as linear correlation. Okay, and if there is a relationship but not both the variable are not strongly connected, then it is called as non-linear correlation. Okay, that means the relationship is present between the two variable, but the relationship is non-linear. Okay, so this is this theory of positive and negative correlation, then simple, partial, and multiple correlation, and linear and non-linear correlation. Now I am going to focus on this. Am I audible? Sir, reply द्या कारण की मला कळत नाही.
Yes, sir.
Yes,
Okay,
sir.
so there are three important methods of finding the correlation. Okay, we saw the three different types of correlation, but now there are three important methods by which you can find out the degree of correlation present between the two variable in your research. Number one is scatter diagram method. Number two is graphic method, and number three graphical method, and number three is coefficient of correlation method. First two methods are qualitative methods, right? That means they just tell you that the two variable are related or not, but they do not give any quantitative measures to you. Okay, so they are qualitative method. But if you want to find out the exact degree of correlation in percent, then you have to go for this coefficient of correlation. So I'm going to explain both this method quickly, and then in this lecture I'm only going to focus on the coefficient of correlation. Right. So number one is scatter diagram method. In this method, you simply put dots on the graph on the x and y axis, and depending upon the pattern of dots obtained, you conclude whether there is a relationship present between the two variable or not. So it is the simplest method of finding the correlation present between the two variable, and you can use this for any kind of problem like this one. The problem is in an experiment, data recorded on two variables. In an experiment, data recorded on two variables such as plant height and number of flower per plant are given in the following table. Determine the correlation by scatter diagram method. They have given you the name that you have to find out the method you have to find out the degree of correlation by using scatter diagram method. And this is the data which is provided in two column table. Number one is plant height and number two is number of flowers, right? That means this data is all about, suppose the plant having 18 centimeter height, it will going to have 20 flowers. The plant with 25 centimeter height will going to have 33 flowers. Plant with 20 centimeter height will going to have 26 flower, and the plant which have 13 centimeter height have 15 flowers. Okay, so table is just showing the number of the height of the plant and number of flowers. If you just observe this table, it is quite difficult to say that whether the two variable are correlated or not. The plant height and number of flower are correlated or not. It is very difficult to say anything regarding about correlation by just observing the data, because sometimes data will be too large, okay, and multiple factor will be there. In that case, it becomes very difficult to make any conclusion, right? So now we have to decide whether these two variable are related or not by using scatter diagram method. काय वाटतं तुम्हाला हे रिलेशन असेल की नाही, प्लांट हाईट अँड नंबर ऑफ फ्लावर्स, असं बघून तर कळत नाही. बट व्हेन वी ड्रॉ स्कॅटर डायग्राम विल गोइंग टू गेट क्लिअर. बॉटनीवालं कोणी असतं का, नेचर म्हणजे जनरली असतं काय रिलेशन प्लांट हाईट अँड नंबर ऑफ फ्लावर्स? येस ऑर नो? 100%. 100%. Okay, so if plant height increases, the number of flower will going to increase by some degree. Okay, and if plant height decreases, number of flowers, so our answer will be there is a correlation. Okay, so now we will going to check by scatter diagram method. Just draw two axes, x-axis and y-axis. Take one variable, generally independent variable on x-axis and dependent variable on y-axis. In the both the variable, independent variable कुठला आहे, plant height or number of flowers? What you say? इंडिपेंडेंट कुठला आहे जो आपल्याला एक्स ऍक्सिस वर घ्यायचं?
Number of flowers will be variable and plant height will be fixed.
Yeah, that means independent is plant height. Plant number of flower depend upon plant height, but plant height is not depend upon number of flowers. So independent, that is plant height will be on x-axis and number of flower will be on y-axis, and then you have to just give these dots. Dot कसे द्यायचे? For example, last plant have height 23 centimeter and number of flower is 30. So just locate 23 on x-axis, draw a perpendicular imaginary line, and then from y-axis draw a parallel line 30 from 30. And when these two lines meet, you have to give dot. Okay, so dot will be on the point where we match 23 on x-axis, right, and 30 on y-axis. The dot will representing 23 and 30. Similarly, if the observation, the plant have 25 centimeter height and 25 flower, in that case, the line will be like this, 25 from x-axis and 25 from the y-axis. The point will be at this point, at this position, right? So in this way, you have to give all the points on the graph or on the diagram, and then with the help of pattern of dots obtained, you can easily make conclusion about what the degree or type of correlation. कसं करता येईल? If the line is moving from bottom to top, that means असा pattern आहे. If the height of the dot is continuously increasing in this fashion, you can say that both the variable are positively correlated. If you get opposite line, that means line pattern is decreasing from y to x axis like this. Okay, just observe my cursor. If line is like this, in that case, you can say it is negative correlation. That means one increases, other decreases, or one decreases, other increases. If the line is like this, you can say it is negative correlation. If the line is like this, you can say it is positive correlation. And if the dots are spread on all over the graph or diagram, you can say that there is no correlation. That means variable x and y is non-correlated. Okay. Another thing is that if you draw a straight line from the center of the graph and all the points come near to the line or on the line, in that case, you can say there is a perfect positive correlation. And if line is coming from y to x axis and all the points are close to the line, in that case, we can say that it is a negative correlation. Okay. So on the basis of this scatter diagram, now the conclusion is that both plant height and number of flowers are having positive correlation. Is it clear to all? Yeah, scatter diagram method is the simplest way, but in this method you only get the idea about whether the correlation is present or not, but quantitative value will not going to get from this method. Okay, the next is graphic method. It is similar to the scatter diagram method. I'm not going to discuss in detail. Okay, so the problem is from the following data, determine whether there is any correlation present between the two variable or not. Don't variable A, variable 1 and variable 2 in rows. And the observation is January, March, May, July, September, November. They have given month and maybe anything, humidity or say soil pH or anything, I don't know. Okay, but the data is like this. The name of month, then variable 1 is increasing like this or behaving like this, and variable 2 is like this. Okay, now from this data you want to find out whether there is a relationship present between variable 1 and 2 by using graphical method. Then simply you have to draw the graph. Okay, just take the observation like January, March, May, July on the x-axis. Okay, and then locate the minimum value of the observation. Minimum value is 4 and maximum value in this case in both this variable is 8.02, right? So depending upon the minimum and maximum value, take the scale on y-axis. So my scale will be 2, 4, 6, 8 because my minimum value is 4 and maximum value is 8.2. And now just plot the line diagram. Okay, variable 1 for January, value of variable 1 is 4. So I have given dot on 4. Okay, this green line represent variable 1 and red line represent variable 2. You can give multiple line on same diagram if there is more than two variable. Okay, in some of our experiment we may plot 50 to 60 line on the same graph depending upon your data. Okay, so in this case green line for variable 1, red line for variable 2, right? Now for March, variable 1 having value 6.01 somewhere at this point, and variable 2 have value 6.89, right? So now in this way you have to just give the spot, then join them with the help of straight line by using scale. And once you join the dots, then you can find, you can easily observe the pattern and then you can make your conclusion. For example, when we go from January to March, variable 1 is increasing because, okay, here you can easily see it. Similarly, variable 2 is also increasing. Now from March to May, variable 1 is increasing, variable 2 is also increasing from this point to this point. It is increased, right? Now from May to July, variable 1 is decreasing and variable 2 is also decreasing because point is somewhat lower than this point. Okay, so both decrease. There is no strict pattern, but you can easily observe that both are decreasing. Now from July to September, first increasing, second also increasing. Okay, so with the help of this data, you can say that the conclusion will be on the basis of pattern of line obtained, it is clear that variable 1 and 2 have positive correlation. Okay, आणि मग तुम्हाला असे वेगवेगळे ग्राफ जर मिळाले, दिस इज पॉझिटिव्ह, दोघं जर अपोजिट जात असतील, हा पहिला वाढला दुसरा कमी झाला, इफ द फर्स्ट इन्क्रीज अदर डिक्रीज, असं जर अपोजिट जात असतील तर यू कॅन से दॅट देर इज अ निगेटिव्ह कोरिलेशन. आणि असं असेल काहीच संबंध लागत नाही by just observation, the conclusion will be there is no evidence of correlation in the given data. Is it clear to all? Yes or no?
Yes, sir.
Yes,
Okay,
sir.
so now we are
Yes,
going
sir.
to focus on most important point of today's discussion, that is what? Correlation coefficient. Okay. Is it visible?
Yes, sir.
Yes, so correlation coefficient is also called as small r. The symbol of correlation is small r. Okay, whenever you are calculating correlation coefficient, always remember that you can represent it with the help of small r. Okay, now both the scatter diagram method and graphic method are important. They are easy, but they only indicate the relationship present between the two variable or not. However, they do not provide any numerical value, okay, how much correlation is present, and for that, therefore, degree of correlation can be calculated by using correlation coefficient. It is also called as Pearson's correlation coefficient. Okay, always remember. It is also called as Carl Pearson's, one of the most decorated scientists of statistics. Okay, so the another name of correlation coefficient is Carl Pearson's correlation coefficient. And for manual calculation, the formula is like this. Okay, I'm going to explain the formula now. It is very easy. Okay, but by using this, you can easily calculate the correlation coefficient for your data. Okay, for example, if you want to find out R, and you have in your PhD work, you have two variable X and Y. Okay, X will be anything, and Y. Okay, so both this variable, the data with you, and now you want to check whether they are correlated or not. You can use this formula: summation of X into Y, summation of X into Y minus summation of X into summation of Y divided by small n. Small n is number of observation in your data. Okay, whole term divided by under root first bracket summation of x square. Unmute कोण आहे म्यूट करा. Okay, so whole term divided by under root first summation of x square into into summation of x आणि त्याचा ब्रॅकेटचा स्क्वेअर ह्या दोन टर्म वेगळे आहेत. Summation of x square into summation of x bracket square, whole term divided by same small n bracket complete, and now this bracket will be multiplied by another bracket. Just add y, summation of y square minus summation of y bracket square. इथे चूक होते मुलांकडून. Summation of y bracket square divided by small n bracket complete. Okay, प्रॉब्लेम घेतल्यावर एकदम इझी होईल हे. फक्त एकदा फॉर्म्युला बघून घ्या. Now the problem is, find out the correlation coefficient from the following data. प्रत्येक स्टेपला वापरू शकता इन युअर रिसर्च. खूप जास्त सिग्निफिकन्स आहे कोरिलेशन आणि रिग्रेशनचा. अँड आय एम एक्सप्लेनिंग टू यू व्हेरी क्विकली. सो इफ यू आर एबल टू ग्रास्प इट, इट विल बी व्हेरी गुड फॉर यू. ओके, सो नाव सेम घेतला आपण डेटा. Plant height on x-axis in the first column and number of flower on in the second column. Now we know that these two variables are related, but we want to find out degree of correlation. Now by just draw or just write formula here. Okay, एकदा formula लिहून झाला की now you going to get what will be the requirement of formula. First you will going to need x into y. तुम्हाला पहिलं काय लागणार आहे, x into y ही टर्म लागणार आहे. सो डेटामध्ये ऍड करा कॉलम, नेक्स्ट कॉलम ऍड करा एक x into y, हे दिसतंय तुम्हाला इथे. फर्स्ट सेकंड थर्ड फोर्थ फिफ्थ कॉलम ऍड केला मी x into y. काय करायचंय, 18 मल्टिप्लाय बाय 20 360, 25 into 25 625, 25 into 33 825. In this fashion you have to find out x into y values in the last column, right? And then just make summation of x into y. It will be 17,532. ही झाली तुमची पहिली टर्म. फॉर्म्युल्यातली तुम्हाला ही टर्म मिळाली. यू हॅव गॉट द फर्स्ट टर्म, राईट? समेशन ऑफ एक्स इंटू वाय. नाव द मायनस समेशन ऑफ एक्स इंटू समेशन ऑफ वाय. मग समेशन ऑफ एक्स काढून घ्या. कुठला आहे समेशन ऑफ एक्स? पहिल्या कॉलमची सम. यू हॅव टू जस्ट make summation of first column, plant height 18 plus 25 plus 25 plus 32 plus 35 up to last observation, that is 23. So summation of x is 543. ही टर्म पण तुम्हाला भेटली. Similarly you have add flower number, so it will going to give you summation of y 603, right? So now you have got both this term, summation of x, summation of y. So now again come to formula. Now you have got the first term summation of x into y, summation of x, summation of y. Now divided by small n. Small n is number of observation. Just count total number of observation from top to bottom. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 60, 70 and 18, 90, 20. So small n will be 20. Okay. So now I have finished with न्यूमरेटर. वरचा पार्ट आपला झाला. आता डिनॉमिनेटर मध्ये हा हे सगळं झालं. डिनॉमिनेटर मध्ये अंडर रूट काय आहे? समेशन ऑफ एक्स स्क्वेअर. एक्सचा स्क्वेअर करून त्याची सम. मुलं काय करतात इथे? 543चा स्क्वेअर करतात. समेशन ऑफ एक्स दिसतंय ना? नाही, इट इज रॉंग. बिकॉज समेशन ऑफ एक्स ब्रॅकेट स्क्वेअर असेल ना इथे, summation of x bracket square, then you can add 543 square here. But in formula they have given summation of x square. म्हणजे पहिल्यांदा x मधल्या कॉलम नंबर 1 मधल्या प्रत्येकाचा स्क्वेअर करायचा आणि त्याची सम करायची. किती किचकट आहे बघा. x स्क्वेअर ची सम आहे, x चा स्क्वेअर नाही. ओके, तुम्हाला अगोदर x चा स्क्वेअर करून मग त्याची सम करायची. समेशन ऑफ x चा स्क्वेअर करायचा नाही. ओके, सो फॉर फाइंडिंग धिस व्हॅल्यू मला काय करायचं आहे, आणखी एक कॉलम ऍड करून घ्यायचा, x स्क्वेअर, दिस वन. So 18 square is 324, 25 square is 625, 25 square is 625, 32 square is 1024, 35 square is 1225. Similarly, you have to add all the squares and then make addition. It will going to give you summation of x square, which is 15,923. And the last calculation will be summation of y square. Okay, you have to just make the square of all the y observation and then add them. It will going to give you summation of y square 19,563. So I have got 5 values: summation of x, summation of y, summation of x square, y square, and x summation of x into y. Now I can put all in my formula. बघा इकडं. Now you can add this. Summation of x into y, the value is 17,532. For this, I have added 17,532. Then summation of x is 543. Summation of y is 603. Right? Divided by small n is 20. Whole term divided by under root. Summation of x square is 15,923 minus summation of y square is 19,000. Sorry, minus summation of x 543 square divided by 20. This bracket is complete. In the last bracket, summation of y square, that is 19,563 minus summation of y 603 square divided by 20. You have to just add all the values in the formula and then step by step solve it. First, 543 into 603, it will be 3,27,429 divided by 20. Similarly, the square of 543 is 2,94,849. 603 square, 3,63,609. Okay, so I have make the square in first step. In the second step, just do division. Okay, so 3,27,429 divided by 20 will be 16,371.45. Similarly, the answer for the this division is 14,742 and 18,180. Now in the next step, do subtraction. So in upper part, 17,532 minus 16,371 will be 1160.55. Under root, whole term divided by under root, the first subtraction answer will be 1180.55 and into this will be 1382. Now just do multiplication. So the answer will be 16,032, I think. So do the multiplication. Wow, sorry, 1 lakh or 16 lakh 32,169.403. Right? Now take the square root. The answer will be 1277. And in the last, you have to just do the division. 1160 divided by 1277. The answer is 0.90. Okay. I forgot to tell that correlation coefficient value range from 0 to 1. कधीच 1च्या वर जाणार नाही, कधीच 0च्या खाली येणार नाही. हा, 0च्या खाली येऊ शकते, 1च्या वर जाणार नाही. राईट? ओके, 1 r इज इक्वल टू 1 मीन्स 100% कोरिलेशन. एक्स जर 10 वाढला तो y पण 10 वाढतोय किंवा त्याच प्रोपोर्शन मध्ये वाढतोय. इट इज कॉल्ड ॲज परफेक्ट कोरिलेशन अँड इन दॅट केस द व्हॅल्यू ऑफ r विल बी 1. r ची व्हॅल्यू 1च्या खाली आली, 090, 080, 070, 060, म्हणजे तुम्ही म्हणू शकता 90% 80%, 70%, 60% नातं आहे, रिलेशनशिप आहे. आणि ती जर -1 कडे गेली, 0 to -1, म्हणजेच निगेटिव्ह कोरिलेशन आहे. रिलेशन आहे पण निगेटिव्ह आहे. ओके, सो इन दिस केस द कन्क्लुजन इज देर इज व्हेरी हाय डिग्री ऑफ काय पॉझिटिव्ह कोरिलेशन बिटवीन प्लांट हाईट अँड नंबर ऑफ फ्लावर. किती हाय डिग्री? 90% रिलेशन आहे. तुम्ही तुमच्या थिसिस मधला कुठले पण 2 व्हेरिएबल कोरिलेशन करून कनेक्ट करू शकता अँड नो वन विल गोइंग टू टेक डाऊट ऑन युअर डिसिजन इन दॅट केस. तुम्ही जर हे तुम्ही स्वतः म्हटल्या theoretically yes, I am thinking that both the variable are related, no one going to believe you. But in your PhD viva or in your thesis or in your paper, if you prove that they are related with their correlation or this coefficient, no one will going to question you. समजतंय का? आवाज येतोय?
Yes.